Right. So now we move on to next part about the bioinformatics analyst for the PacBio long read sequencing. Ting Ting, please check it.
Yeah, okay. Let me share my screen. Okay. Yeah, I guess you can all see my screen, right?
Yes, I can see your screen now. Thank you.
Okay. Okay. So, good evening, everyone, and welcome again to for joining today's webinar. And so, I would like first to thank Ray for introducing the advances in sequencing with PacBio Hi-Fi reads, both on the data quality and the cost. And in the following 30 minutes or so, I will tell you about how you can achieve those high quality results using the bioinformatic resources we offer here from PacBio. So here I'm showing you again the four primary applications using Hi-Fi technology in the microbiome space. So on the left, you can see microbial whole genome sequencing. So you do a culture and you perform isolated sequencing and then full-length 16S sequencing, which allows you to get to species and strain level identification. And then the last two are both related to metagenomics, which is the shotgun approach. And in the following time, I will talk in detail on specific data analysis approaches for each of these four applications. So let's start with microbial whole genome sequencing. And with regard to the microbial genome assembly, we have this workflow, which is called microbial genome analysis workflow. This workflow is specifically developed for microbial genomes assembly. And with this workflow, you will obtain not only the chromosome assembly, but also the plasmid assembly simultaneously. And the output sequence has been formatted for NCBI submission, so you don't do any modifications on your sequence. You can just submit the output results directly to NCBI. And in addition, you can also specify the base modification detection if you would like to have that information. For example, you can specify to obtain 6MA or 4MC information using this workflow. And this microbial genome analysis workflow is available in our software SmartLink. So SmartLink is our official software, and it is implemented with a graphical user interface. So this will make things much easier if you are not familiar with the command lines, especially you're a wet lab researcher and wants to analyze the data by yourself. And here I'm showing you how this analysis workflow looks like in SmartLink. So it is just a push button workflow. And all you need to do is to indicate the data set you want to analyze, and then to determine whether you want to also detect this base modification or the motif-based motifs. And finally, you just hit this start button, and the analysis will perform automatically. So to show you the performance of this workflow, we prepared 96 libraries from 24 distinct bacteria relevant to food safety and infectious diseases. Those different bacteria has a wide range of GC content, genome size, and complexity, and each with 4 replicates. And we then sequenced and assembled them using this microbial genome analysis workflow. So here in the top figure, you can see that we have 92 out of 96 microbial genome assemblies with the expected chromosome numbers. And you can also see in the bottom figure that most of the bacterial genomes have the expected number of plasmids. And when we look at the assembly accuracy of these microbial genome assemblies, we found that these assemblies are all around Q50 or so. So that's one of the benefits you can get from using highly accurate Hi-Fi reads. And here I'm showing you a case study that we performed in collaboration with a customer. What they wanted to do is to evaluate the different sequencing technologies and how it works for their antimicrobial resistance, or AMR, detection pipeline. So Here's the result on the contiguity of the assembly using ONT and the PacBio data. So ONT data was shown in blue, and for PacBio data, they used two different assemblers. The first one is our microbial genome assembly workflow in SmartLink. So here we use MA for short, and the other assembler is Hi-Fi ASM. Shown. So the results from these two workflows are shown in different shades of magenta. So here black is the expected number of contacts. So you can see that compared to the expected contacts, PacBio outperforms ONT for most of these bacteria. And when looking at the accuracy, sensitivity, and the precision of that study, you will see that PacBio in magenta here outperforms both Illumina and ONT across the board, especially for accuracy and sensitivity. And then the customer fed their AI algorithms with the whole genome assemblies created with each technology to predict a bacterium's resistance or susceptibility to a specific antimicrobial compound. So as you can see in this table, PacBio data outperforms in agreement and accuracy while showing the least errors. And next, we will move to the full-length 16S sequencing. And we know that all bacteria have a 16S gene, and variation in this conserved gene enables clade identification. So this is a study from our collaborator that they wanted to see the differences between individual variable regions of 16S and how they're able to classify bacteria to the species level. And then they compared these results to those from the full-length 16S that include the whole set of V1 to V9. So their results showed that since the human gut can harbor a broad diversity of bacterial clades, so only full-length 16S species can provide unbiased resolution of all the species that may be present in the microbiome. And furthermore, they were able to identify SMP locations through facing full-length 16S gene sequences generated for each individual isolate. So here you can see that x-axis denotes position along the 16S gene, and y-axis denotes individual isolates clustered based on their inferred phylogeny. And the dark blue region, so these dark blue regions indicates the location of a polymorphism. So as you know, the short-read 16S sequencing looks at only the V3 to V4 region. So they found that 91% of cultured isolates are not distinguishable because of intragenomic SNPs. But on the contrary, full-length 16S with Hi-Fi sequencing can provide a wider range of polymorphisms for species and strain level identification. So to help you achieve the species and strain level identification, we developed an ASV-based workflow to analyze full-length 16S Hi-Fi sequencing data. So this is the overview of the PB16S NF pipeline, and you may find the detail in our GitHub page here. So you can just click this link to get to our GitHub page, and we have detailed documentation to tell you how this workflow how to install this workflow, and we have also demo data set for you to test it, to play it around. And here I would like to mention, so after taxonomy classification, this pipeline will generate an HTML report and a visualization to summarize the key results and statistics. And most importantly, the output from the pipeline is fully compatible with KIME2. So we ran this workflow across different types of samples and found high percentage of reads assigned at the species level. Although here you can see in magenta that the relatively low number of reads assigned at species level for some complex environmental samples. And this also highlights the need for sample type specific databases, which may improve taxonomy resolution. So we rerun the taxonomy classification against the MIDAS wastewater database. And then these two numbers um, increased to 85% and 91% respectively using the MIDAS wastewater database. So the next application is read-based profiling and classification. And as Ray has just mentioned previously, the goal is to identify the species present in the sample and estimate their relative abundances. But there are several challenges associated with profiling and classification. And the most important one is that a lot of the methods for long reads are still being developed. And so the best practices and how these methods performed was unclear. And there's also a difficult balance between precision and recall. So that is how we can minimize false positives while also being able to detect species at very low abundances. So to address these questions, we performed a comprehensive benchmarking study of profiling and classification methods, especially for long-read shotgun metagenomic dataset. And this study has been published in the paper shown on the right in 2022. So here I'm showing you the 11 methods we tested, and we were able to identify 3 top performing methods, including sour mash, diamond and Megan LR, as well as bug-seq. So these are there are still some differences between these methods. For example, sour mash is chimer-based, whereas diamond and Megan LR is based on protein alignments, and bug-seq is based on nucleotide alignments. And one thing I want to point out about the diamond and Megan LR workflow is that in addition to generate a taxonomic profile, it also simultaneously creates a functional profile. That's currently the only method that's capable of doing that simultaneously. And we have built workflows for both sour mash and diamond and Megan, and you can also check them out in our GitHub repo. So now let's jump into some of the results of these 3 methods. So you can see the numbers of species detected at each of these different abundance categories. So in the top row, we have ultra low abundance, and towards the bottom, we have higher abundance. And what I want you to focus on first are these methods in gray. So which are the optimized filtered methods tuned for high precision. And we can see that we detect 150 to 55 to 223 species based on these filtering criteria. And in white, so these 2 columns, I'm showing some unfiltered results from these different methods. And what you can see is those numbers jump up quite a bit, up to greater than 7,000 species with no filtering, and which you will notice most of these assignments are happening in the ultra low categories. And based on our study, we've quickly identified most of that is noise. So this demonstrates that the importance of using this threshold for internal filters in these programs to obtain high quality confident results. And also here, I'm showing you some of the results from the artificially downsampled datasets using the Diamond and the Megan workflow. So we have 4 smart cells of data down all the way to a 48 plex. So this represents a 90 gigabase of data down all the way to 0.5 gigabase of data. And from comparing the results, we not only see the same number of species, but also really consistent relevant abundances across total data. So this really opens the door for using shallow shotgun sequencing to generate highly accurate taxonomy profiles. And as I mentioned previously, the Diamond and the Megan workflow can simultaneously output functional annotations in addition to these taxonomy profiles. And you can see that approximately 92% of the HiFi reads received at least 1 functional annotation. And in total, over 67 million functional annotations were assigned across all the four databases. So this again shows the capability to build a highly accurate taxonomy as well as functional profiles using hi-fi reads. And here's another example showcasing that hi-fi data is rich in functional information. And in this table, you can see 5 human fecal samples. The mean hi-fi rate length is about 9 kilobases. So this means you can detect 8 to 9 genes in one hi-fi read, even without assembly. So in a scenario of genotyping your metagenomic samples, you can just go directly with the gene annotation, and there's no need to assemble hi-fi reads. And one exciting thing is that only 4 long-read methods available during our benchmarking study mentioned previously. However, over the last maybe 6 to 7 months, 6 new methods focusing on long-read taxonomy profiling and classification have appeared. So this really suggests that the research community is recognizing the power of long reads. So now let's move on to the last main topic of today, which is metagenome assembly. And the goal here is to assemble a representative genome for each microorganism present in the microbiome sample. And we refer to these genomes as MAGs for metagenome assembled genomes. So there are quite a few challenges here. And first of all, there are multiple species present in the microbiome sample. And second, these species are present in uneven and unknown coverages. And finally, there are repeat regions and intergenomic overlaps that occur across species, as well as between strains of the same species. But the good news is, hi-fi reads can really help with this problem. So in the beginning of 2022, we published a new assembly algorithm called hi-fi ASM Meta. So this is specifically tuned for the hi-fi reads. And in the publication, we demonstrated that hi-fi ASM Meta produces more total max and more importantly, more circular max than any of the other methods that are currently available. And in January 2024, another powerful tool called Meta MDBG is published in Nature Biotechnology. And we have done some work to compare these two assemblers with others. And our results showed that these two assemblers are currently the best practices for assembling MAGs. So just to give an example how hi-fi ASM Meta looks like, and here's an assembly graph from a human gut microbiome sample. And look closely, you will notice there are actually 75 circular contexts here. And these all range from about 1.2 to 6 megabases in size. So these are complete microbial genomes that come strained from the assembler. But you will also see that there are plenty of linear contexts here. And this is the reason why we would need post-processing pipeline to identify and extract these additional high-quality marks from this complex set. As such, we developed a hi-fi MAG pipeline, and this pipeline takes assembled context and outputs the high-quality MAG sequences, metadata, and visualizations. Now, the version of this pipeline is 2.0, which implements completeness-aware binning strategy. As a result, version 2 shows not only an increase in total max, but also an increase in single contact max comparing to the previous version. And so in this example, we run hi-fi ASM Meta and the hi-fi MAG pipeline on the same human gut microbiome sample. So we were able to recover 189 high-quality marks and classify them into 154 species. One of the interesting things is that 14 of these marks cannot be classified to the species level, and these are likely to represent a novel diversity in the human gut microbiota. In addition, there are 19 species that are represented by 2 to 3 marks, implying there's a strain level variation among them. And so here shown on the bottom, we obtained two representative marks from the same species and align them to the genome taxonomy database reference. So you can see in these dot plots figures here, there is still some structural variations across the genome. And that's even true if we compare the two marks to each other. So remember, these two marks are from the same species, and we can still observe those structural variations. And in the following time, I'm going to show you two pilot projects that we collaborated with two different groups. So one is the Banfield lab at UC Berkeley, and the other is from DSMZ in Germany. As you may have been aware, that soil is so far the most difficult environment sample because it has the most diversity of microbial species, and many of them are in very low abundances. And therefore, it is very difficult to assemble all of the organisms that occur within those communities. So we performed a deep sequencing on three different types of soil samples, including wetland, woodland, and grassland, using our Revio system. And we generated 3 to 4 smart cells on Revio per sample. We assembled them using our two current best assemblers, Hi-Fi AS Meta and Meta MDBG. And then we conducted binning and QC using like the post-processing using our Hi-Fi Mac pipeline that's available on our GitHub. And finally, we also performed downsampling experiments to understand various data levels and how many marks you can expect from those data. So here in this table, you can see we have about 200 gigs of data for the Banfield dataset. We have 280 gigs for DSMZ1 and 150 gigs for DSMZ2. And here's an example sampling graph from the wetland soil of the Banfield example. And you can see here a lot of circles that represent a complete chromosomes. And we actually have about 360 approximately of these circular contexts within this one sample. And these are all greater than 1 megabase in size. And we have one that's even super large here, which is a 13 megabase genome. So looking at the actual number of marks recovered, we're getting hundreds of thousands of total marks from all of these different samples. And many of them are single contexts, and as much as 50% of the total marks are actually high quality marks. So in this study, high quality means greater than 90% completeness and less than 5% contamination. And medium quality is 50% complete and less than 10% contamination. And more importantly, the marks consist of very few contexts. So the medium contact number for all marks are 4, and then for high quality marks, the medium is 1 contact. So you can see we really obtained a very high quality results. And here I'm showing the number of marks versus the total data generated for those 3 samples in comparison to the Zymo-Chill matrix standard, which is a human fecal reference sample. With the Zymo sample, so this one, we have reached saturation by sequencing on 4 Revio cells to successfully characterize every single species of the strain. With the soil samples is a different story. And you can see that they're still very linear. So we're not accomplishing saturation. It means if you add more data, you probably will get more high quality marks. So the Banfield sample is still ramping up, but it's probably getting close to saturation. We just don't know yet. And the reason for this is the different species diversity in each sample. So the species diversity of human gut reference is relatively low. It's only about 600 or so. but the Banfield wetland soil is medium, and the two DSMZ samples are in high and very high diversity. And so here we have provided the latest publications on long-read metagenomics. So the top one is a review paper covering both PacBio, Hi-Fi, and ONT sequencing. And the bottom one is our preprint discussing the current best practices for Hi-Fi metagenome assembly. And finally, we have all these digital available resources for your reference, and you can access all these resources at our website, pacb.com. So with that, I want to thank you for your time today, and we're happy to take any questions.
Thanks a lot, Ting Ting. Very nice talk.